Well, thank you for the opportunity to speak today. I'm a final year PhD student in the Manila Lab at the University of Cambridge, and I'm going to be talking predominantly about one project specifically, but also other projects that I have been working on utilizing volumetric electron microscopy. And I think it's very important for us to say that this is a very collaborative project with groups around the world, and none of this would have happened without sort of the great community of the imaging world and specifically in the volumetric electron microscopy field as well. So the airway epithelium serves as the first line of defense against inhaled pathogens, bacteria, and toxicants. And the key process to this is called mucociliary clearance. Mucus is released from goblet cells that trapped particles, which are then subsequently removed due to the well-coordinated beating of the motile cilia. And these are tiny nano-beating machines located on the apical surface of multiciliated cells within the airway. Now, recent studies employing single-cell RNA sequencing has transformed our views of the airway epithelium, demonstrating that basal stem cells have the ability to differentiate into novel cell types and cellular transitional states. And so what you can see on sort of the left hand of your screen is that sort of traditional landscape where you have a basal cell, ciliated cell, a goblet cell, and just a club cell. But what single cell has done has provide a much more complex view of the airway epithelium, where we have rare cell types like neuroendocrine cells, hybrid cells between a goblet and a ciliated cell. And whilst our transcriptional understanding of these differentiation trajectories are well characterized, how these genetic changes result in alterations to the cellular architecture, and by that I mean changes in organelle topology and interactions within the cell, remains unknown. And I think this lack of knowledge limits our ability to fully comprehend how diverse cellular populations coordinate with each other to not only drive intercellular processes within the airway epithelium, but also maintain tissue homeostasis and prevent disruptions in response to disease and toxicity. And so previously, the lack of technological advancements couldn't provide us with the ability to answer this question. However, as we all know, recently, a group of technologies called volumetric electron microscopy has emerged. And this is a beautiful group of technologies which essentially provides us the ability to visualize whole cells in 3D at a nanoscale within the native tissue environment. And specifically, the technique that I'm going to be talking about is a scanning electron microscopy, volumetric electron microscopy technique called focused ion beam scanning electron microscopy, or FIBSIM. And I'll go into more depth in later about how it works and with regards to sample prep, etc. Now, for the preprint paper that I will be presenting today, I'm focusing specifically on the differentiation of a basal stem cell to a multiciliated cell. And this process is also known as multiciliogenesis. Now, during this trajectory, basal cells first progress to deuterostomal cells, which is almost an intermediate state between basal stem cells and multiciliated cells. And in this intermediate state, hundreds of basal bodies are generated within the cytoplasm and transported to the plasma membrane for docking. And as you can see, this is a structure of a single motile cilium. And so we have the axoneme, which is very important for ciliary beating. And then you have the basal body, which is located at the base of the cilium. Now, the basal body has multiple accessory structures, one of them being the transition fibers, which is essential for basal body docking to the apical membrane. The other is the basal foot or basal feet, which are essential for coordinated ciliary beating. And lastly is the ciliary rootlet, which is a striated fiber that emanates from the base of the basal body. And its structure, its structure and its function in human airway cells is still quite elusive. And so, how do we obtain the 3D view of the airway epithelium? Now, what we do is that we obtain biopsies from healthy individuals, and we can actually acquire biopsies also from patients with disease and toxicity. And we grow them on an in vitro culture system called the air liquid interface culture system. And this recapitulates sort of the in vivo settings of the airway. Now we embed these cells in a resin block, and for imaging, we cut a slice, image that slice, cut another slice, image it, etc., all the way through the resin block, which ultimately provides us with a 2D stack of images that we can then analyze for volumetric segmentation and overall 3D reconstruction. And so this was one of the first data sets that I was sort of tasked with segmenting. And we can see we have the outer cellular space in white, and we're going deep into the cell. And again, I was tasked to identify every single structure that I could find and segment it. And it's just by this sort of view, it provides sort of the beauty of what volumetric electron microscopy can give us. It gives us this unbiased perspective of the tissue and cellular organization of that airway epithelium. Now, we've also developed a sort of manual and AI combined segmentation pipeline in order to analyze these data sets. And the reason it's combined is because some structures require manual segmentation, such as the rootlets, which I'll delve into later on, and then others can sort of you fine with automated segmentation. And so utilizing Empanada and the Napari plugin, which is developed by Conrad and Kedar Narayan and published in Cell Systems, we combine that with 3D mod package from iMod to create this sort of combined hybrid segmentation pipeline that we've published from the lab in a methods chapter. And so this is the 3D reconstruction of the previous volume that I showed. And this is actually all manual segmentation before we sort of delved into the hybrid approach between manual and AI segmentation. And so it took a very long time, but I hope we can all agree it's quite beautiful at least. And what it does, it really provides us the opportunity to study the cellular architecture of multiple different cell types of the airway epithelium. And so we've got a multiciliated cell, which is fully differentiated. We have a deuterosomal intermediate cell, and we also identified a goblet cell. And because we were interested in just multiciliogenesis, we were specifically looking at the comparison between the deuterosomal cell and the multiciliated cell. And we were able to identify the deuterosomal cell based on the identification of a deuterosome, which is this fibrogranular material. And we can see we have migrating centrioles coming off, and we have the 2D SEM plane here. And I think maybe we can see some docked axonemes and some migrating centrioles. But with multiciliated cell, we can see this aligned basal feet all facing in a similar direction or the same direction, which is indicative of full differentiation as well as axonemes, of course. And I think just by this slide, we should appreciate the sheer beauty of volumetric electron microscopy, as it reveals that cells not only just undergo sort of transcriptional changes, but undergo extensive changes within its nanoscale architecture across multiciliogenesis. And it provides us with this rich sort of ability and information to try and uncover new mechanisms which are driving these changes within the subcellular architecture. And so one of the changes that we found interesting was changes in nuclear architecture and perinuclear organelle localization. And so cross differentiation, the basal stem cells sort of elongate along the apical basolateral axis. And also what we found in basal stem cells is that the invaginations of the nucleus are filled with endoplasmic reticulum and mitochondria, indicative of mitochondria-nuclear communication. Now, this is in vast contrast to differentiating cells, whose nuclei is characterized by the presence of large lipid droplets around the nuclear envelope. And this is actually consistent with work from the Crick, which said that multiciliated cells require sort of rapid metabolic rewiring towards fatty acid beta oxidation during both differentiation and repair. Now, a significant difference in cellular architecture that we also observed was in the distribution and the topology of the mitochondria. And we show this by quantitative morphological analysis of individual mitochondria. Now, volumetric EM, whilst a beautiful technique, is still quite low throughput, especially with segmentation with regards to the pipeline. And so we also integrate super resolution imaging in conjunction to validate our findings. And you can see that this is a super resolution micrograph of a multiciliated cell on its side. And we can see that the mitochondria is incredibly tubular and elongated, but also seems to be very close to the surface of the cell as well. And this is consistent with its role in ATP generation for ciliary motility. Now, these structural changes are then supported by single-cell RNA-seq analysis from the human lung cell atlas core, where we see an increased expression in mitochondrial dynamics, specifically proteins related to mitochondrial fusion in multiciliated cells. Additionally, we also saw a marked increase in genes related to mitophagy in multiciliated cells. And this may be due to the need for mitochondrial quality control to support high energy demands, remove ROS-damaged mitochondria, and facilitate apical clustering for efficient ATP delivery. And this is actually backed structurally by interactions between autophagic organelles and mitochondria as well, which is incredibly interesting. But our most interesting finding was that we found that the mitochondria form contacts with the rootlets that emanate from the basal body close to the apical surface. Now, as you can see, the rootlets are separate from each other. They don't intertwine with each other, making us to assume that these are meaningful contacts. Now, the rootlets were either found to wrap around the mitochondria or even have a kiss and touch physical interaction as well. And we actually found across 4 differentiated cells that 50% of the rootlets make at least one contact with the mitochondria in human airway multiciliated cells. Now, this interaction between the mitochondria and the rootlets has been shown over 60 years ago in marine animals. However, this is the first to be shown in the human airway cells. Nonetheless, what this observation shows us is that this is a conserved organelle cytoskeletal interaction. And what this observation also then led us to do is to study the molecular and cellular architecture of the rootlets, utilizing a combination of advanced imaging technologies, and then understand how its architecture contributes to the function of rootlets and their contact with the mitochondria. And so we had individual questions that we wanted to answer. What are the morphological characteristics of individual rootlets? When do rootlets form during multiciliated differentiation? What are the rootlets' cellular architecture? What are rootlets' molecular architecture? And how are rootlets linked to the cellular base? And most importantly, what is the function of rootlets and their interaction with the mitochondria? And just for time, we're just going to focus on those highlighted in orange. So, What's the cellular architecture of rootlets? On the screen, you can see an example of a rootlet and the cilia from a side-on view, where we have the axone, we have the basal body, we have the basal feet, and we have the rootlet and the corresponding micrograph. Now I'm hoping this works. Over here, we have a manual segmentation of a rootlet that I segmented. And this really gave me the ability to assess the morphological characteristics of individual rootlets from multiple differentiated cells. And so this took a very long time. I think there was around 499 rootlets that I had to manually segment and go through. And that's why I'm a big fan of 3D Mod. I think it's a great tool to use for complex sort of structures such as rootlets. Um, but yeah, it really provides us this ability to look at the individual morphological characteristics of individual rootlets. There we go. Now, our 3D segmentation of rootlets from multiple cells showed that they form this asymmetric dome-like architecture. And this is very dissimilar to what has been shown in lower organisms and in cyclin cells, but also in primary ciliated cells. I know this is a segmentation from the mouse epididymis dataset I've also been looking at. And you can see that rootlets here are extremely different in cellular architecture between species, suggesting that maybe not only is the architecture difference in different cellular contexts, but also the function. And so it raises that question about, okay, how is the subcellular architecture and organization of rootlets linked to the functionality of that specific cell type? And so we wanted to first quantify this asymmetric dome-like appearance. And to do so, we determined the anterior and the posterior of the end of the cell. And we used the basal feet as a proxy for cilia beating directionality. From there, we then mapped the rootlet's endpoint relative to the start point, giving it either a positive or negative curvature relative to the forward effective stroke, or in other words, the direction of cilia beating. And what we found is that the rootlets converge to the back end of the cell relative to cilia beating directionality, suggesting that the intracellular architecture of the rootlets may be linked to cilia beating functionality. Next, we wanted to assess the molecular linkage between the rootlets and the cilia base. And so to better resolve the link between the rootlets and the basal body, thanks to the MRC toxicology EM facility, we employed high pressure freezing and free substitution electron microscopy. And what this revealed was a bridging complex characterized by fibrils emanating from the proximal end of the basal body. Now, to better understand the molecular composition, we then use super resolution nanoscale mapping to do so. And in this process, we use a reference marker, in which this case is POC1b, which marks the basal body and is well characterized. And then we use candidate proteins which have been previously implicated to interact with CROC or rootletin, which is the main component of rootlets. And what this allows us to do is to build a map, and the first nanoscale map of the basal body and basal body rootlet linker. And so what you can see, we have, let me just get my laser out. We have CEP250 here, and then we have rootlets over here, croc, and which is croc. And then we have other elements of the basal body. But there's still a gap between the two, suggesting no direct interaction between CEP250 and croc. And so we wanted to look at other components, and I found that LRRC45, which has been shown to be a component of central linker in cyclin cells, not only localizes to transition fibers, but also at the linker region between the basal body and the rootlet, forming this filamentous fiber. In fact, there was overlap between both LRRC45 and CROC, suggesting a molecular interaction. But these are all proteins which have been previously implicated to interact with CROC and rootletin in different cellular contexts. And we wanted to identify new components of this molecular bridge. I then examined CROC2, a paralog of CROC and CEP250. Now, single-cell RNA-seq analysis shows that CROC2 has a similar expression profile to both CROC and CEP250, suggesting it to be a bona fide component. And this is consistent with super-resolution imaging, which showed that CROC2 forms a ring-like structure close to the basal body, with a short filament partially overlapping with CROC, suggesting a molecular interaction. And so the localization of LRRC45 and CROC2 suggests that the basal body linker region is much more complex than previously demonstrated in previous studies. Now, thanks to Esben Lorentzen and colleagues in Denmark, we then used AlphaFold to predict interactions between these key proteins. And we found that CEP250 and CROC2 were predicted to interact. And additionally, LRRC45 was predicted to interact strongly with CROC and CROC2. Now, taking all of this together, the AlphaFold and the super-resolution imaging and the nanoscale mapping, this suggests that these proteins may form a higher order assembly at the basal body rootlet linker. Now, what's the molecular architecture of the rootlets? Now that we looked at the cell architecture and we looked at how it was linked, the molecular architecture was next. And so to do so, we utilized 2D STED or stimulated emission depleted microscopy plus deconvolution to attain a pixel size of 15 nanometers. And so this is a confocal view of the rootlets. And so this is a side-on view of the multiciliated cells. Imagine that we have the cilia here at the top, the apical surface, and here's the basolateral surface, and we have the rootlets forming, or half forming, this sort of dome-like architecture. And here we have, of the same cell, 2D STED plus deconvolution. And what we can see just by this is that the N and the C terminus form of rootletin, of CROC, form this striated, repeated pattern. And what this suggests, after doing some measurements, we found this not only to be consistent across individual rootlet fibers, but it suggests that rootlets assemble in a polar manner. So, the big question, what is the role of the rootlets in human airway multiciliated cells? Now, we first wanted to understand rootlet dynamics during ciliary beating. And to do so, an airway cell line expressing EGFB CROC was generated using the piggyback integration system. And this was thanks to Chris Godber here, who's a co-author, joint first author on the preprint, who did the sort of GFP work and also the knockout future experiment works. So obviously a shout out there. And so employing super resolution live cell imaging to attain both high temporal and spatial resolution in a temperature and CO2 controlled manner, we found that the rootlets seem to undergo this consistent oscillatory movement with frequencies similar to those in ciliary beaten, suggesting that they have a counterbalancing role to motile cilia forces transduced. So to understand the role of the rootlets, we generated a CROC knockout cell line thanks to Chris using CRISPR-Cas9, followed by clonal selection to generate a single knockout population. And what I found was that the multiciliated cells differentiated normally, suggesting that they are not essential for ciliary assembly and stability. And this is very different to what has been shown in primary ciliated cells. We also found that the croc knockout cells exhibit similar ciliary length relative to the controls, as well as similar beating frequency. Now, we also then wanted to look at how knocking out croc or root-letting disrupts the localization of proteins at the basal body linker region. And what we found is that croc knockout has no effect on croc2 localization. But what we see with LRRC45 is that whilst LRRC45 localizes to the transition fibers, as we go deep down into the cell, it doesn't localize to the basal body linker region, suggesting that there may be two separate molecular pathways playing a role in the recruitment of LRRC45 to both the transition fibers and the basal body rootlet linker region. And the recruitment to that region may be dependent on croc and root-letting. And this is just a table summarizing the recruitment and, well, those findings. And so we looked at ciliary beat frequency, like I said before, and there was no effect on ciliary beat frequency, nothing significant between the two conditions. We then wanted to assess the effects on ciliary beating coordination. And by doing so, we analyzed the basal body and basal feet alignment in both conditions. Now, typically, this is performed with transition electron microscopy, where we look at the basal body and the basal feet across multiple cells, but that's low throughput. And instead, I adapted a MATLAB script, which is previously published by our lab, which allows us to analyze a high number of cells from multiple replicates. And so as you can see in the control, we have this alignment of the basal body in red and the basal feet in green, and it's super aligned rotational polarity. But when we look at the knockout, we can see that that's lost and that polarity is lost and there's disorganized, suggesting uncoordinated beating and indicating that rootlets are central for maintaining beating coordination. Next, I want to assess the effects of ablating rootlets on mitochondrial organization. And so using super resolution imaging, I isolated multiciliated cells and found that whilst there was no clear signs of mitochondrial fragmentation observed in the croc knockout cells, unlike the control, where the mitochondria was concentrated apically, in the knockout cells, we found that the mitochondria were more distributed and diffuse along the apical basolateral axis, thereby suggesting that rootlets play this novel role in facilitating mitochondrial positioning apically. Now, we then wanted to look at the effects on mitochondrial metabolism, and we developed an assay using high-resolution respirometry. And so first we measured the basal level respiration of the cells at basal state. And then we added an uncoupling agent called FCTP, which drives the electron transport chain to its maximum capacity. Lastly, we added a complex 3 and complex 4 inhibitor to measure oxygen consumption attributed to non-mitochondrial sources for our calculations. Now, whilst the basal level respiration was similar between both conditions, the maximum electron transport chain capacity was significantly reduced in the croc knockout cells. And this indicates that there's an impaired capacity to support oxidative phosphorylation, which is the main pathway for ATP generation, when there's a lack of rootlets, thereby resulting in lower ATP generation and highlighting a novel role of rootlets in regulating mitochondrial ATP generation. And so overall, we used this unbiased segmentation approach to generate a nanoscale map of the airway epithelium. After identifying multiple key structural changes across cellular states, we focused our analysis on rootlets and their interactions with the mitochondria. And this led us to discover a novel form of communication between cilia, rootlets, and mitochondria, which are required to regulate ATP generation. And so I just quickly also want to talk about the CellMap project, which has been in conjunction with HHMI Genelio, who have provided us with these rich data sets, 100 by 100 by 100, I think, which are online. Definitely have a check out of it. It's absolutely beautiful. And so with regards to this project, I've been going around different data sets, trying to segment as many cells as possible, rare cells, whatever, from the airway epithelium. And, one of those examples is the pulmonary neuroendocrine cell, which you can see over here, which is a rare cell type. And then another sort of reconstruction I've been working on is this mucociliated transitional cellular state. And the reason why we call it, well, it's been identified by single cell many years ago. And the reason why it's this sort of mucociliated cell is because it has the axonemes, but also has these mucus granules, suggesting a transitional state between both ciliated and goblet cells. And potentially a evidence, and we have sought super resolution as well back in this, but evidence of trans differentiation. And I think from a disease perspective, this is very interesting and is one that needs to be further sort of interrogated and analyzed, and I hope by those in the lab they will. But also sort of shows how we can now expand volumetric EM with super resolution, with other sort of assays into sort of the disease world to have a better understanding of disease and not just fundamental cell biology. And with that, I just want to say thank you specifically to my PI, Dr. Manella, and as well to Chris, who's the PhD student and co-author on this paper, and an extensive thanks to the lab and all the collaborators who have made this, possible. So check out our preprint as well. We have additional bits of structural changes and stuff on the rootlets that I think is really cool. Obviously, I'm biased. And yeah, thank you for listening. And I'm also looking for postdocs. So if anyone wants to hire me, please do reach out. Think of this as like the first line of interview. But yes, thank you so much.